This is our project for AP Biology with Nick Andrews, Amanda Ewer, Connor Nolan, and Tyler James. We will be demonstrating chemical signaling in the cell.
Alright, so here we're going to start to show you how ligands from the outside, the exterior of the cell, outside of the cell membrane, can transport their messages through the cell membrane, through what we call the G protein, to eventually in the long run activate and phosphorylize different types of molecules inside the cell. So in this case we're going to be activating glucose to exit the cell. That's going to be our end result here. So on the outside of the cell we have two different types of ligands. We have our lipid-philic and we have our lipid-phobic ligands. Now, since lipids play a huge crucial role in the structure of the cell membrane, lipid-phobic ligands cannot attach or enter the cell membrane. So in order to get their messages to the interior of the cell, they're going to attach onto what we call the G protein. So there we have our lipid-phobic ligands attaching to our G protein. Then, on the other side of our G protein, we have our subunits. So the message is going to transfer through our G protein to this furthermost subunit, which we call the alpha subunit. So then we are going to take our alpha subunit here, and it's going to get transported through the interior of the cell membrane to what we call the adenylyl cyclase. From here, we have our ATP, which gets formed. Oh, yeah, okay. And then we have our ATP that gets formed due to the energy that is released from our adenylyl cyclase. And then as a result, some of this ADP is going to lose two of its phosphates. And as a result, we are going to have cyclic AMP, or CAMP, take its place and appear. So now that we have these different forms of energy on the interior of the cell membrane, they are going to come up to what's called the protein kinase. Now, the protein kinase is consisted of two different proteins. We have the catalytic proteins and we have the regulatory proteins. So now our cyclic AMP, or CAMP, is going to go and attach itself to the regulatory proteins as part of the protein kinase. So here we go. Our CAMP is giving the energy off to the protein, and as a result, this kinase is going to split apart. So now that we have our regulatory protein split up our protein kinase, so now our catalytic proteins are going to take some of that energy that was released from the adenylyl cyclase and take that ATP and going to become what we call active. So now we have active proteins. Now these active proteins are going to go and attach to enzymes inside of the cell. In this case, we'll have phosphorylase. So then they come here, they attach to our phosphorylase, and our phosphorylase then becomes active. So then that can come over here to our glucose, which then triggers the activation of our glucose, which allows glucose to be released and be sent through the cell membrane. And that's how chemical messengers from the outside of the cell transport and signal their